Regenerative medicine is the future of healthcare. It will complement the functions of lost, damaged, or failing tissues and organs by restoring and regenerating cells and tissues outside the patient's body. It will realize treatments that until now were impossible. But we must remember that all treatments come with risks. To undergo treatment through regenerative medicine, there are three options. Publicly insured treatments. Approved by the Ministry of Health, Labor and Welfare, these treatments are covered by health insurance. This is the healthcare we normally receive. Clinical researches. Patients take part in research to verify the safety and efficacy of treatments. Treatments become eligible for public insurance coverage once they are approved, but this may take a long time. Private clinical practices. Patients undergo treatments that have not or not yet been approved by the Ministry of Health, Labor and Welfare upon concluding agreements with medical institutions. For example of benefit, these may include treatments that have been approved overseas but not in Japan. However, one thing you must be aware of in particular is that the high cost of a treatment does not necessarily mean greater safety or efficacy. Doctors and patients will need to discuss and agree to the details and costs of private clinical practices, but sometimes the expected results are not attained, or treatments have been known to deteriorate conditions. To avoid such situations, it is important to grasp a good understanding of your treatment and whether safety and efficacy can be ensured. Make sure the doctor gives you a thorough explanation with documents clarifying the specific details, including risks and costs of private clinical practices. For example, make sure that the authorities have been notified of the treatment as a due process and that it will be executed accordingly. And check the expected results and dangers associated with it. You should check with your doctor if there is anything you don't understand. The U.S. Food and Drug Administration, FDA, offers the following tip-offs to protect consumers from problems associated with health foods. You should refer to this list when checking information on healthcare too, to see if any of these descriptions fit. The Japanese Society for Regenerative Medicine has opened a portal site for sharing information on regenerative medicine and offering consultation services for patients. You can use this site to gather information. Choosing the right treatment is a major decision in life. You must exercise the utmost care.
Hello everyone, I am Yoji Sato from Japan National Institute of Applied Sciences. I am also the Vice Chair of the Database Committee of the Japanese Society for Regenerative Medicine. Today, I would like to introduce two legislation systems for regenerative medicine in Japan and a nationwide database for regenerative medicine, which is called NRMD. This is a disclaimer. This slide shows you the regulations for regenerative medicine and cell therapy in Japan and the United States. As you can see, there is a remarkable difference between the two countries. In Japan, cell therapy products for marketing are regulated by Pharmaceuticals and Medical Devices Act, PMD Act for short. Cell therapies provided in hospitals and clinics are regulated by Act on the Safety of Regenerative Medicine, RM Safety Act for short. In contrast, a common regulation applies to both in the United States. The scopes of the RM Safety Act are medical practices using specified processed cells without marketing authorization, which include out-of-pocket treatments and non-commercial clinical researches. The scopes of the PMD Act are manufacturing and marketing of products for regenerative medicine or cell therapy or gene therapy by firms, which include commercial clinical trials of regenerative medical products. Let me briefly explain this track. The RM Safety Act consists of three major components. First, the Act obligates hospitals and clinics to submit their plans. Second, it enables commissioning cell processing to licensed enterprises. And third, it obligates cell processing facilities to notify or obtain license. Regarding cell processing, from a viewpoint of patients, the quality of the processed cells should not be different between the two legislations. So, common good practices for cell processing apply to both tracks. We call the common good practices GCTP, which stands for Good Gene, Cellular, and Tissue-Based Products Manufacturing Practices. So the RM Safety Act shares some similarities to European and British systems for use of unlicensed cell therapy products, which are called specials and hospital exemption. In these systems, evidence for the efficacy is not required. Also, no product license is required, but a manufacturer license is necessary, which look similar to the RM Safety Act. The RM Safety Act has contributed to protection of the public health since its implementation. As you can see in this slide, in order to prevent future adverse events, the government can arrest medical practitioners who conduct cell therapy without notifying the authorities. But the Act is not perfect yet, and there are some concerns. As Nature Journal pointed out, there have been some concerns about the qualifications and independence of the committee that approved treatments under the RM Safety Act, because rules about conflicts of interest on the committee have been lax. The ministry instituted policies to prevent such conflicts, but even with fully independent committees, clinics can shop around for the answer they want. So the government is considering extra fixes, such as requiring training to make the committee system better. So let's move on to PMD Act. As you may have already known, in the PMD Act, about 6 years ago, we introduced a step of conditional and term-limited approval, which accommodates early practical application of regenerative medical products. If clinical data are likely to predict efficacy and confirming safety, the product may be eligible for the conditional and term-limited approval. So far, 11 regenerative medical products, including 2 in vivo gene therapy products, have been approved for marketing. We are also expecting 2 more products to be approved soon. As you can see, 3 or 4 of them are conditional and term-limited approvals. A problem in Japan is that the quality of data from non-commercial clinical researches under the JSRM Safety Act has not been necessarily compliant with good clinical practices under the PMD Act. So a lot of data cannot be used directly for marketing authorization. This is our challenge. In addition, after the conditional and term-limited approval, we need to further confirm the safety and efficacy during the post-marketing study. So collection and analysis of the real-world data are another challenge. To solve these problems, the government decided to develop a nationwide database for regenerative medicine. Please take a look at the introduction video.
November 2014. Revised Pharmaceuticals and Medical Devices Act shows a whole new pathway of regenerative medical products which enables fast approval with a small number of cases and randomized controlled trials that are not always necessary. According to the characteristics of cell-based products, when its safety is confirmed and the efficacy is assumpted, regenerative medical products can be approved with conditions and time limit. As a result, the products can be delivered quickly to the patients, and the cost of pre-marketing phases can be decreased. However, the efficacy of products should be evaluated in an adequate manner in its time-limited marketing phase, of course, which means high-quality post-marketing studies and real-world evidence play an extremely important role in a conditional, time-limited approval system. The National Regenerative Medicine Database, the NRMD, is designed to support the research and development of regenerative medicine in the phases of clinical trials and post-marketing studies. The NRMD is developed by the Japanese Society for Regenerative Medicine, the JSRM, in collaboration with the regulatory authorities in Japan. Data registration items common to all diseases are constructed in collaboration with PMDA, and data items for individual diseases are added following close consultations with relevant academic societies. With this scheme, you can construct an efficient marketing platform in Japan. The data are registered in the Computerized System Validation Protocol, pursuant to the Ministerial Ordinance on Good Post-Marketing Study Practice. Additionally, NRMD is compliant with the CDISC standard, which has the potential for future application to international research and development. The EDC package, which NRMD adopted, has been used in 64 countries, 14 languages, 12,000 sites, and assures real-time backup in numerous sites. The use of this series of databases facilitates the easy utilization of clinical data in pharmaceutical regulations and in the construction of high-quality datasets, ensuring authenticity even in clinical research where this is not required. Moreover, as a system to gather real-world evidence, NRMD is recommended by the Ministry of Health, Labor, and Welfare for its quality. The NRMD has 2 registration phases. One is NRMD-CR, used for clinical researches, and the other is NRMD-PMS, for post-marketing studies. Registration of data in this comprehensive database from both clinical researches and post-marketing studies allow us to easily perform translational and reverse translational researches. As a registry system recommended also by the government, the NRMD makes your development highly rational and efficient and realizes smooth entry to the Japanese market. Delivering regenerative medicine safely, effectively, and quickly. JSRM, the bridge to the innovation in Japanese regenerative medicine.
Since regenerative medicine covers a variety of diseases, a wide range of expertise is required when determining the input items for post-marketing surveillance. In order to accommodate products that target various disease areas, the member societies of the Japanese Association of Medical Sciences organized the Regenerative Medicine Registry Committee to quickly determine the data entry items for each product. The committee is composed of members dispatched from each of the member societies of the Japanese Association of Medical Sciences. At first, a marketing authorization holder of a regenerative medical product makes a request to the Secretariat of the Committee, which is the Japanese Society for Regenerative Medicine, to formulate items for the product. In turn, the Secretariat asks appropriate committee member societies to form a working group for the product, taking the marketing authorization holder's opinions into consideration. Then, the working group for each product discusses and decides the input items. Also, the regulatory authority will be invited as an observer. This slide shows you how the registered data are expected to be utilized. For NRMDCR, for non-commercial clinical researches, data quality is assured by performing computerized system validation in compliance with good post-marketing study practices. For non-commercial clinical researches that allow for a control group, data from the control group can be registered with the same quality. Data from previous control groups can be used as historical controls in subsequent non-commercial clinical researches or post-marketing surveillance. In cases of products for which it is difficult to have a control group, smooth product commercialization without relying on randomized control trials can be supported by setting up a PMS-focused R&D design. This is what it looks like in the illustration. On the left is the CR data. On the right is the PMS data. So the data can be exchanged with the same quality. NRMD is a database constructed in collaboration of PMDA and JSRM and operated by JSRM. However, it is not the obligation of the marketing authorization holder to use the NRMD. In fact, some marketing authorization holders use their own database or the database for a nonprofit or academic organization, such as FormsNet 3. But the regulatory authorities in Japan are asking companies to cooperate in the use and data accumulation of the NRMD, considering future development of cell and gene therapy products. If you are interested in our database, please check our website or just email us. Thank you for your attention.
Hello, my name is Hideki Okano, the president of JSRM and also the professor of the Keio University School of Medicine. It's my great pleasure that I can make a talk in these great opportunities. Today I'm going to talk about IPS-based regenerative medicine for the spinal cord injury. Okay, so it has been long believed that adult mammalian central nervous system does not regenerate upon injuries, as Dr. Ramoni Kahar had indicated long time ago. This is Kahar's dogma, but we wanted to challenge this dogma with the power of the stem cells. Neural stem cells are somatic stem cells present within the central nervous system. They can give rise to a wide variety of cells in the central nervous system, neuron astrocyte and oligodendrocyte. And they are present in the fetal brains abundantly that can be expandable in vitro. They are also present in adult brains on only very small levels, but they are not easy to expand. Later, it turned out that fetal-type neural stem cell can be induced from the pluripotent stem cells, such as ES cell and iPS cells. Okay, so this is a roadmap of the neural stem cell transplantation for neural stem cell. We have working the stem cell transplantation for spinal cord injury more than 20 years before the born of the iPS cell technologies. First, we have tried the rat fetal neural stem cell transplantation for the spinal cord injury model in the adult rats. Then we planned the clinical trial for the human fetal neural stem cell transplantation for the patient with spinal cord injuries. But we noticed that big difference between rodent and human being in terms of function and anatomy of the spinal cord. So that's why we started to develop a non-human primate spinal cord injury model using a common marmoset. Then we transplanted a human fetal neural stem cells. It nicely worked. So then we planned the start of the clinical trial. But however, Japanese guideline on the use of the human stem cells for clinical research released in 2006. This did not allow us to the use of the aborted fetal derived cells and human ES cells. This guideline is no more present, but we had to identify some sources for the neural stem cells. And then 2006, groundbreaking papers as published from Shinya Yamanaka's group named iPS cell technologies. Now the ESL access can be obtained from adult fibroblasts. So in collaboration with Dr. Yamanaka. So we started this project. Now first we induced them into the neural stem cells through neuralized embryo bodies. This neural stem cell gave rise to neuron astrocyte and oligodendrocyte, and then transplanted into the mouse spinal cord injury model. So this is contusion injury at the thoracic level. So the mouse showed a strong paralysis in the hind limb. But when we transplant the iPS the neural stem cells, it showed a nice function recoveries. And we also noticed iPS neural stem cells provided maximal therapeutic outcomes when they transplanted within a few weeks after the injuries. And then the human iPS, using human iPS cells, we indeed induced them into neural stem cells and transplanted into the NOD skid, spinal cord injury model, and also common marmoset spinal cord injury models. This is a controlled marmoset spinal cord injury model. So the marmoset cannot move, and so their leg, and so and also their forelimbs. And however, by transplantation, the subacute phase, so marmoset show that remarkable function recovery like this. So in the function of both forelimb and hind limbs. Okay, then what is the action mechanism function recoveries? We found that transplanted iPS neural stem cells nicely differentiated in oligodendrocyte. This is the level GFP in this case, and so GFP positive myelin as present on the both sides of the Caspar positive as the Rambier node, indicating they contribute in the myelinations. And also iPS neural stem cell different into a neuron and then made synapse with the host neurons. As a presynaptic or postsynaptic mechanism, they are involved in the synaptic relay of the disrupted neural circuit. In addition, they produce some pleiotropic factors. Okay, so then how is the procedure of develop the clinical grade iPS neural stem cells? We tried 3 methods, protocol A, B, C. In protocol A is the prolonged embryo body formation, neurosphere formation, all through the floating cultures. Protocol B, embryo body, rosette formation in mono cultures, and then sphere formation. Okay, and protocol C, embryo body, and so rosette formation in the monolayer, then sphere cultures, and monolayer culture again. Okay, so then we transplanted these neural stem cells obtained 3 different methods. And so transplanted into the brain of the NOD mouse and NOG mouse, and also transplanted in the spinal cord injury model of the NOD mouse, and performed the histological evaluation 12 to 26 weeks after the injuries. Okay, here is the result obtained. Nice survival and differentiations. And protocol BNC, some extramedullary outgrowth could be observed occasionally. So protocol no tumor density and no abnormal cells. Protocol and BNC, although there's no sign of the tumor density, but so there's some extramedullary outgrowth. So this cannot be used. But protocol C, some ossification, even some ossification. So protocol A seems to be the best protocol. According to this, we have developed the procedure and so for the clinical grade iPSDM neural stem cells. So on the side, we got the clinical grade iPS cell in KO. We expand them by 2 passage and twice, and then neural embryonic body, and then the neurospheres. And the 4 passage, and we prepares the final product. Okay, so by using the sterility and toxin, the mycoplasma, and so whole genome sequencing, transcriptome, and also in the iPS cultures or the flow cytometry and the karyotyping and cell cycle growth, and so also immunocytochemistry, and also we perform examination tumor density assays by transplanting into NOD mouse. Okay, so then we clear these issues. And then this is a working flow of the clinical trial. From the side, we have got the clinical grade iPS cell. We induce them neural stem cell. And after quality check in vivo, in vitro, now we have Rosenstock neural stem cells. When appropriate patient recruited in Keio University Hospital, age over 18 years old and 14 to 28 days after the injury, and so they should be complete injury age 11A. So then informed consent is obtained. We make the wake-up culture and transplant into the interregional injection, 2 million cells for individual patient. And we keep the rehabilitation and observation 1 year after the transplantation in collaboration with the medical centers. And so this is class 1 regenerative medicine protocols. So it's an open labels, and so single group assignment for patient, and the neurological status age A and 2 or 4 weeks after the injury, age over 18 years old, transplant iPSDM neural stem cell, interregional injection to the spinal cord. And so, these are the primary outcomes, and mainly the safety issues. Anyway, so our protocols, clinical protocols are class 1 regenerative medicine protocol according to the Japanese Act. So the Act on the safety in regenerative medicines. And so now we are now ready to start. And so we wish we could have started December 1st last year, but so due to the COVID-19 outbreak, so it's start is somewhat postponed. But we wish to start soon when the COVID-19 outbreak comes down. Now the future indication is chronic spinal cord injury and also stroke. And for this, we already have preclinical POC has been obtained. And also in the future, we wish to apply for the multiple sclerosis by supplying the myelin-forming oligodendrocyte and Huntington disease and other neurodegenerative disorders. Okay, so now this is iPS cell in the clinical trials, approved one and ongoing. So there's 13 cases across the world, at least in the end of 2020. 11 out of them is now currently ongoing in Japan. So the PI is all the PI of the Japanese clinical trials, member of our JSRM. We hope to contribute to human health with the power of the stem cells. Okay, thank you very much for your attention.
Yasuji Watanabe, Managing Director of Department of Regenerative Medicine and Cell and Gene Therapies at the Japan Agency for Medical Research and Development, abbreviated as AMED. I appreciate the opportunity to introduce the mission of AMED and the work that we have been doing to promote the development of regenerative medicine. First, I'd like to outline the operational structure of AMED. AMED is a funding agency established in 2015 by the Japanese government to support the seamless application of basic research to the clinical setting in the field of medicine and effective establishment and maintenance of an environment for medical R&D. AMED does this by consolidating related budgets from three ministries: MEXT, MHLW, and METI, involved in research of medicine in the Japanese government. From April 2020, AMED has entered its second 5-year period since its establishment. And as an agency, we have 6 integrated projects centered on modalities indicated in gray, and disease priority areas indicated in blue that span across the projects. This is so disease research can be promoted across the integrated projects and thus managed flexibly. Of the 6 integrated projects, our project is the Regenerative Medicine and Cell and Gene Therapies project, indicated in red, and I will explain the project in the next slides. This figure shows our funding programs across different stages of research to bring promising technologies to the clinical application stage. This structure helped in promoting seamless or encompassing support for regenerative medicine, including laying down the foundation for active regenerative medicine research and the promotion of regenerative medicine techniques such as disease-specific iPSCs for use in drug discovery. From left to right, the figure shows the stage of research progression. The leftmost part of the figure is basic and applied research, indicated in green, supported by MEXT. Moving to the right, this is followed by preclinical research, indicated in pink, supported by MHLW, and then clinical research and trials that will lead to the clinical and practical use of the research outcome in question. Commercialization of the research outcome is indicated in blue, supported by METI. In this slide, we show the detail of the project. Research Center Network for Realization of Regenerative Medicine, indicated in green. I will describe in more detail about 2 important programs indicated by the red boxes in this project in the next slides. This slide shows the details of the program. The Center for Clinical Application Research on Specific Disease and Organ. In this program, 11 center facilities for clinical application research were established, and each facility was designated a core organ or disease. 10 facilities utilize iPSCs, and 7 centers have already started clinical studies. This slide shows The second program, the acceleration program of intractable disease research, utilizes disease-specific iPS cells. This program supports a wide range of research from basic to preclinical research of disease-specific iPS seeds. In this program, 19 projects that target different diseases, such as neurological, respiratory, muscular, and bone and cartilage disease are ongoing. The established disease-specific iPS seeds are stored in a cell bank managed by the Bioresource Center, BRC, at Riken. As controlled samples, Saira at Kyoto University established iPS seeds from 100 healthy donors and deposited them into the cell bank at BRC. Through these efforts for seamless research funding, the drug candidates indicated on the right of the slide have already entered the clinical research stage. Next, I will introduce the pink funding areas that focused on the later stages of clinical research and clinical research and trials. There are two main projects running in these stages: the program for practical application of regenerative medicine and the project to promote the foundation for practical application of regenerative medicine. The program for practical application of regenerative medicine supports the execution of doctor-led clinical trials and clinical research. About 24 clinical research and trials are currently being funded through this program. In this slide, we show the graphical abstract of project to promote foundation for practical application of regenerative medicine, which promotes the practical application of regenerative medicine in Japan, in which university hospitals, national research institutes, corporate groups, etc. take part in a coalition called the National Consortium of Regenerative Medicine. The core of this national consortium is the Japan Regenerative Medicine Society. In cooperation with the National Consortium for Regenerative Medicine, the regenerative medicine clinical research model hospitals that have extensive experience in clinical research and doctor-led clinical trials in regenerative medicine support collaboration with medical institutions across Japan that would like to implement regenerative medicine clinical trials and research. Lastly, this slide introduces the key program with the project focused on developing basic technology aimed at industrialization of regenerative medicine and gene therapy indicated in blue. This project supports all phases of research from basic to clinical trial and research, with commercialization of promising seed technologies as a key focus. The 4 key programs within this project are the commercialization of promising seed technologies that will lead to new regenerative therapy products, indicated in the top left. Establishment of stable raw material supply systems for regenerative medicine at the top right. Development of efficient processes for manufacturing regenerative therapy products based on the concept of quality of design, QBD, indicated in the bottom right, and the use of regenerative therapy products in drug discovery through development of platform technologies indicated in the bottom left. As you can see from these 4 programs, this project focuses on key technology advances for the commercialization and implementation of regenerative therapy products. In these ways, we promote basic research on regenerative medicine, non-clinical and clinical research and trials, development of foundational manufacturing technology, and establishment of necessary foundational structure for research. To end this presentation, I'd like to show you a visual representation of life 20 years from now, based on feedback from roughly 5,000 experts across various fields of science. As part of the outreach program, we use this image as a tool for science communication to the public, and a broad goal as AMED and related organizations strive for better future for all. Thank you for your attention.
Hello everyone, my name is Takanori Takebe from Cincinnati Children's Hospital and Tokyo Medical Dental University. So today I'm going to talk about organ research going around my 4 different laboratories in Cincinnati as well as in Japan. So my lab is really interested in studying the tissue organogenesis on the focus on the liver. So this is a live imaging analysis of the liver tissues in an animal model. And as you can acknowledge this beautiful imaging, the tissue structure is very sophisticated, having multiple different cell types. So the questions we are dealing with is to how we can grow the human organ tissues in a dish for the study of human development and disease. So having human iPS cells or ES cell available, we are currently now utilizing the technologies termed organoids or miniature organ technologies to try to tackle these problems. So organoids is defined by a couple of reasons. For example, organoid is essentially resembling the normal organ structures or miniature organ structures, having, for example, the multicellular compositions in the tissues. But not just the existence of the multi-cell populations, but they need to structurally organized in a way we see our normal in vivo structures. And because of these 2 definitions, we are able to see the functional emergence in our organoid-based system. For example, in the liver case, hepatocytes is secreting number of key proteins that is induced in our organoid model systems. So my laboratory is spending decades of years to study how we can do this by learning the normal organ formation process or organogenesis process. So translating the knowledge of organogenesis into culture principles so that we can direct the stem cells to grow more complicated organ systems with a focus on the liver. So we first time in 2013 reporting first generation of the liver system comprised of multiple cell types. But over the years, we have been updating multiple times so that we can incorporate additional components into the liver organoid system. For example, last year, we have been able to generate macrophage bearing liver organ model model system so that we can model inflammation or fibrosis pathogenesis. And up in the middle, we also have been able to generate liver blood vessel networks populated in the liver organoid system. So if we transplant this vascularized liver organoid in an animal model, we can initiate the functional blood perfusion, as you can see from these pictures. On the far right, so this is most recent one, updated ones, having the connection to the neighboring organ system. For example, when you look at the liver in our body, they are connecting to biliary systems or pancreatic systems and connected to the duodenum. And this connection is really vital for the liver health and homeostasis. So we kind of decided to tackle this question of the connection to the liver. So in 2019-2021 papers, we are describing the methods to generate the connection to liver and biliary as well as pancreatic to duodenum population. So having that connections, we are able to model the connection failure, for example, biliary atresia or primary sclerosing cholangitis model system predicted in our organoid model system. But going forward, we are primarily, as a clinician scientist, interested in translational efforts of these, complicated organoid model system. In other words, can we make an impact on the current human healthcare with the use of the human organoid model system? So to address these questions, we are currently thinking three major directions we can potentially envision in a shorter term future. So because of the stem cell technologies, we are now able to form the organoid from health and disease conditions. And from humans particularly is exciting because we can replace the animal experiments to study the human specific questions associated with the disease progressions. But beyond that, for example, when you use the patient's organoid model system, we can potentially select the better medications to find out more effective treatment tailored to the patient's specific condition. So organoid can serve as a diagnostic tool to predict the medical treatment protocols. When we provide the healthy organoid for the pharmaceutical industry, they may be really acknowledging the supply of organoid because it's really hard to predict, for example, the toxicity or side effects of developing drugs. So organoid for the drug development application really benefit the pharmaceutical industry in time-consuming laborious drug developmental processes. And finally, if we can use a healthy organoid and transplant into the patients with disease conditions, we can potentially envision the therapeutic use of the organoid for other treatments to the devastating condition. So just to summarize, we think that organoid medicine application is something we are potentially applying in our future. So I'm going to showcase the three major directions with each example with a focus on the liver organoid samples in the next slides. So this is the first application as a diagnostic examples of the patient-specific organoid samples. So this is the patient's reports. So we are collecting the patient's stem cells from the 82-year-old females diagnosed as a dementia. The problem was she was repeating the pneumonia by repeated infections. But the problem was when she's treated with the medication to combat the infections, liver toxicities is emerging, and which is really concerning. So they need to stop the treatment by antibiotics and rather stabilize the liver toxicity conditions. So to potentially help selection of the antibiotic treatments, we decided to make an iPS cells and making organoid and combine with the, for example, lymphocytes or neutrophil fluctuations and see if we can model the drug-induced damage that was observed in the patients. And this really efficiently predicting the patient's condition So as you can see in the middle panel, we have identified ampicillin as well as levofloxacin was extremely toxic to these patients' organ samples with the immunity components, whereas the other class of drugs is not so much toxic to these patients' conditions. So, and in the later clinical care, we also know that these low predicted samples, antibiotics, is actually not really toxic to these patients, kind of validating our results. And finally, we can also apply these organoid for more mechanistic studies. And for example, when you target the IL-1 beta cascades, we can prevent the cellular toxicities. So kind of suggesting if we can use the anti-IL-1 cascades neutralizing the antibodies, maybe we can treat the conditions when she has a toxicity. So that is sort of diagnostic applications. Secondly, if we can provide the organoid for the pharma industries, we can select the better, more safer compounds in an efficient manner. So we have published a paper, so I cannot really go through in details, but we have developed a 384 well-based reproducible organ screening platform to study drug-specific toxicity mechanisms predicted in our organoid system. So finally, we have also transplanted the liver organoid into the disease animal model and showing you significant improvements after transplantations from the liver failure conditions compared to the sham operated mouse. And this is kind of quantifications of our organoid benefits for the lethal liver failures. So taken together, we think that organoid medicine has an impact on the three major directions, and that was published in multiple publications. So hope we can convince you that organoid has a potential for making an impact on the clinical care. With that, I would like to thank all my colleagues in my lab, as well as collaborators and funding agencies. And we are welcome for the collaboration with the multiple external parties.
Hello, I'm Yomabuchi. I'm very grateful to the organizers for giving me an opportunity to present this work on the regulators of senescence in the human mesenchymal stem cell by Freeze-05. As you know, the mesenchymal stem cell, which are referred to as the stromal cells, hold a considerable promise of the cell therapy. However, long-term culture of the MSU lead to the cellular senescence, meaning that the cell loses their therapeutic potential. Here, we found that Freeze-05 play an important role in regulating the senescence in the human mesenchymal stem cell. Let me explain to you in detail our study. We have previously identified cell surface marker specific to the human mesenchymal stem cell, LNGFR and Cy1, CD271 and CD90. These markers allow the isolation of the mesenchymal stem cell of the human bone marrow. You can see on this FACS profile that the mesenchymal stem cells are enriched in the co-positive fraction of the LNGFR and Cy1. These cells exhibit self-renewal and multilineage potential. Therefore, we concluded that the LNGFR and Cy1 co-positive cells are mesenchymal stem cells. Using these markers, we were able to perform the single-cell sorting of co-positive fraction and follow up their growth in culture. Interestingly, we observed that the clone had a very different growth pattern. Based on these growth rate, we defined three different classes of the mesenchymal stem cell: rapidly expanding cell, slowly expanding cell, and moderately expanding cell. This classification probes the heterogeneity in the MHC population. Our analysis shows that REC have not only a higher proliferation, but also the higher the differentiation potential. Another interesting observation, based on the senescence-associated beta-gal staining, was only a few senescence cells could be observed in the long-term culture of the REC, in contrast with others. The smaller size of the cell body in REX groups supported that REX are immature MSC. Based on these results, we started the new study. We decided to take advantage of the differences between REC and other MSC to identify the molecular mechanisms of the regulating the senescence the mesenchymal stem cell. First, we compare the immature REC with the other MSC using the gene expression analysis. And also, we performed the pathway analysis show that these genes are involved in the Wnt signaling. Especially, FRISL5 was found to be the particularly highly expressed in the RECs. Examination of the cell freshly isolated from a bone marrow also shows that FRISL5 was highly expressed in the MHC fraction. FRISL protein, a transmembrane receptor, it's considered that the specificity between a Wnt ligand and receptor complex is determined by frizzled class of receptor. Quantitative PCR and Western blot and FACS experiments show that ROD2 is particularly highly expressed in the RECs, as well as the freshly isolated MHC fraction. Please note that ROD2 is a co-receptor of the FRISL5, which aggregate and activated non-canonical Wnt signal. Consistently, immunostaining confirms that co-localization of the FRISL5 and ROD2. These results suggest that non-canonical Wnt signal is activated in mature human mesenchymal stem cell. Next, we found that the Wnt5A, a ligand of the FRISL5, was highly expressed in the freshly isolated MHC. Moreover, when Wnt5A added to the culture medium of the RECs, their proliferation was increased. This indicated that non-canonical Wnt signal play an important role in immature human mesenchymal stem cell. Next, we wondered about the effect of the decreasing FRISL5 expression in the mesenchymal stem cell. We used the short hairpin RNA to knock down FRISL5 in the RECs. We observed that the reduction of the FRISL5 was associated with the decreasing proliferation. In addition, the morphology of the cell became flattened, similar to that of the senescence MHC. Finally, differentiation assay shows that REC had also lost their ability to differentiate into the multiple cell types. You can see here, when the expression FRISL5 is reduced, REC is a significantly decrease the adipogenic potential. Like other type of the tissue stem cell, MHC show reduce the ability in the long-term culture. We found that long-term culture, the expression of the FRISL5 was downregulated, while senescence markers such as P16 and progerin were upregulated in parallel. Interestingly, the expression of the FRID5A was also downregulated in the long-term culture, suggests that senescence could result from a reduction in the non-canonical wind signal. To examine if the FRID5 related signal was directly involved in the senescence MSC, we knock down the FRID5 in leg. The reduction of the FRID5 result in the increased number of senescence cells and upregulation of the senescence marker. The expression of the gamma H2AX and telomere length are indicator of the cell aging. The knockdown of the FRID5 was upregulated the expression of the gamma H2AX and telomere length was shortened. These data indicated that cellular senescence was accelerated by knockdown of the FRID5. Conversely, what happened if we continue to maintain FRID5 gene expression in the leg? We found that overexpression of the FRID5 allow maintaining the proliferation capacity of the legs in the long-term culture. Let me summarize our findings. Here, we observed that FRID5 is specially expressed in the functional MSC, where it activates non-canonical wind signaling. We have demonstrated by the series of the mechanistic experiments that FRID5 prevents the senescence and prevents the multipotency in the most immature MSC. Furthermore, maintaining the FRID5 gene expression allows the suppressing the senescence in the human mesenchymal stem cell while maintaining their stem cell property throughout long-term culture in vitro. These findings may prove useful in the development of the MSC therapy application. Thank you very much for your attention.
Hello, I'm Akito Hotta from Center for iPS Research and Application at Kyoto University. I'd like to thank the organizer for giving me the opportunity to present our institution's effort towards the clinical application of iPSL. Our institute, Center for iPSL Research and Application, has been established in April 2010. Since then, our institute has been growing, and last year, CIDA Foundation was also established. The CIDA Foundation is to bridge the iPSL technology between academia and industry to advance the iPSL clinical application. Under the strong leadership with Director Shinya Yamanaka, we are working hard towards the mission of our institute. For iPSL-mediated cell therapy application, considering the source of iPSL, there are mainly two options. One is using cells directly derived from patient's own cell, which is an autologous setting. From immunological point of view, this is identical to avoid any risk of immune rejection. However, this approach takes time and cost, hence it's not realistic still to make this approach fully available. In that regard, allogenic cell source from healthy volunteer donors would be attractive alternative to as a source for iPS cells. However, for allogeneic approach, you need to consider how to avoid immunorejection due to the mismatch of HLAs on cell surface. There are multiple reports demonstrating the possibility of avoiding allogenic immunorejection. The main approach would be disruption of beta 2 microglobulin gene to eliminate all the cell surface HLA molecule, or selective elimination of HLA genes, or transgene mediated suppression of immunocell. My group previously reported that selective disruption of HLA-A and HLA-B, but retaining other HLA class 1 molecule, is an attractive way to suppress both T-cell mediated immunorejection and NK cell mediated rejection. With this approach, by matching the HLA-C haplotype, we estimated that roughly 12 lines should be sufficient to cover most of the worldwide population. With this approach, so this is an example of our estimation. So we generated iPS cell from HLA homozygous donors and knocked out HLA-A and HLA-B genes homozygously. We also disrupted the class 2 HLAs by knocking out C22A gene. So only HLA-C is expressed in this case. Before genome editing, this iPS cell induces killer T cell activation or CD4 helper T cell activation. After knocking out of HLA-A and HLA-B, as shown by the yellow box, no killer T cell or helper T cell activation was observed. Also, in the case of beta 2M knockout, CD56 positive NK cell was activated, but our HLA-C retained lines were able to suppress NK cell. Hence, our HLA-C retention approach would be effective to avoid both T cell and NK cell immunity. Since then, we have been optimizing the CRISPR-Cas9 mediated gene knockout protocol, and by using the clinical grade genome editing materials and the electroporator instrument, we have successfully achieved very high double knockout or triple knockout efficiency as shown in this flow diagram. So we are quite confident that with our optimized approach protocol, we should be able to generate clinical grade iPS cells with HLA-AB double knockout or HLA-AB and C22A triple knockout iPS cells. And now we are in collaboration with CYRA Foundation, and we are in the process of generating such clinical grade HLA edited iPS cell. Because iPS cell can be differentiated into many different cell types, so that potential of iPS cell for clinical application is huge. So here are some of the examples that clinical application of iPS cells has been investigated in Japan. And the green boxes indicating the clinical trials already ongoing. Yellow boxes indicate the clinical trials that soon will be initiated, and the gray boxes indicate the planned clinical trials. So in terms of the immunorejection, we think there is no single answer to completely avoid immunorejection. Hence, depending on the cell type and the tissue type to be injected or the target disease, fine-tuning or optimization would be necessary. For example, neuron in the brain, it is more immunoprimarily site, so that the HLA mismatch does not necessarily have a significant major impact. So even HLA mismatch neuron can be survived in the brain. In terms of retina, it's been known that HLA class 1 expression is low and HLA class 2 is almost not detectable. So in this case, HLA class 2 editing may not be necessary. In terms of, in case of platelet, this is interesting, but NK cell can attack HLA new megakaryocytes. However, NK cells do not attack HLA new platelets. So from this aspect, for platelet application, HLA new iPSC might be more beneficial than HLA-C retention iPS cell. In the case of cartridge, again, HLA class 2 is not expressed, so class 2 editing may not be necessary. So depending on your cell type of interest, proper HLA editing approach should be investigated. Here is a summary of my talk today. So optimized electroporation of Cas9 ribonucleoprotein complex gives high genome editing efficiency in iPS cells. With this optimized protocol, we are now working to generate clinical grade iPS cells with HLA-A and B knockout. Still, there are questions that remain. For example, among various HLA editing approaches, which one is the best for clinical application? I think it's depending on the application type, and also side-by-side comparison will be necessary. In terms of genetic tool to induce HLA knockout, so we've been using CRISPR-Cas9 as well as Cas3 system, which is has a very unique system to induce broad genomic DNA deletion. So we are also actively investigating CRISPR-Cas3 system to be used in iPS cell. Of course, safety is the most priority for iPS cell clinical application. So genomic integrity, antigen presentation ability, and the proper animal model to assess the safety and efficacy of iPS cell will be very critical points, and further investigation will be necessary. I'd like to thank my lab members and institutional members who are very actively working towards the clinical application. And so I'd like to thank their efforts, and thank you very much for your kind attention.
Hello, this is Masayo Takahashi from Kobe. I'm now the president of Vision Care Incorporation. The Vision Care is a part of the eye center in this building. One is the research center, and one is City Eye Hospital. And the third one is the nonprofit organization that will take care of the patients called Next Vision. And the fourth part is the Vision Care. And this company has already 2 subsidiary companies called VC Gene Therapy and VC Cell Therapy. In Kobe Eye Center, we are developing various pipelines that arise from the eye center. But main part is the retinal cell therapy development. And our target cell is retinal pigment epithelial cells and photoreceptor cells that consist of the outer layer of the retina. In Kobe, our group performed 3 clinical studies already, and currently we are doing 1 new RPE multi-centered clinical studies. First one was autologous RPE cell transplantation, and the second one was HLA-matched allogenic RPE suspension treatment. And the third one was the retinal sheet that includes photoreceptor cells transplantation. With those experiences, we are now convinced we can make a good treatment, especially various treatments for the outer retinal diseases. In the second clinical study, we've made these real-time immune reaction test called LGIR test and DSA test. LGIR test detects the T cell reaction to the donor cells, and DSA test detect the antibodies to the graft cells. Those tests were very efficiently used in the second clinical study, and we proved that if the HLA was matched, that means HLA effect was removed, the immune reaction was controlled without any systemic immunosuppressive drugs. From this experience, we are now making a HLA partially knockout iPS cells. And here you can see the LGIR test reveal those HLA knockout or shows the defect of the HLA2 expression, even though the inflammatory conditions. We made those various HLA partially knockout iPS cells, and all those cases shows the very little T cell activation, even though co-cultured with the donor cells. So we are thinking suitable HLA knockout, partially knockout iPS cell lines, especially for the RPE. And we are now thinking about the strategy to make all the these treatment, autologous and HLA knockout allogeneic, and RPE cell sheet and RPE suspension, or with allogenic retinal sheet, depending on the cases. It's very important to select the suitable cases for those treatment. And we will utilize fully this special regulatory system for regenerative medicine in Japan. And we will make those various treatment for the outer retinal diseases. With the hospital, we can prepare all the surrounding things about the treatment. Genetic diagnosis or genetic counseling, as we said, immunological test. We can develop surgical technique and device with the clinical team. And also the ophthalmology test should be developed, like adaptive optic camera or polarized light OCT. And also total care of the patient is needed for this kind of new, completely new treatment. Okay. So we would like to prepare all the treatments for autorenal diseases within 10 years. And the vision of the VC- group is total solutions for every patient. Thank you very much for your attention.
Hello, I'm Koji Ito from Saira, Kyoto University. I would like to introduce our project, platforming from human iPS cell toward clinical application. In Japan, total blood donor population is decreasing and much less in the future. Random platelet transfusion or pregnancy can cause allosensitization, leading to generation of anti-HA class 1 or HPA antibodies. By such antibodies, platelet transfusion refractoriness PTR occurs in 5 to 15% of platelet transfusion. These patients require compatible platelets. However, they might not be available for patients with rare types of HA or HPA. For such PTR patients with rare types, patient-derived autologous iPS platelet should solve the problem. Therefore, we performed the first human clinical study. In this single-center, non-blind, non-controlled dose escalation study, we evaluated the safety of autologous iPS platelets as a primary endpoint for anaplastic anemia patients with PTR due to platelet-reactive antibody. Here is the study shown. There are 3 dose cohorts. Cohort 1, 10 billion of iPS platelets. Cohort 2, 30 billion. And cohort 3, 100 billion. Each cohort was evaluated by Data and Safety Monitoring Committee 1 month after administration of iPS platelets. Same procedure was applied for every dose cohort. Primary endpoint was safety, and 1-year follow-up examination started from the administration of the last cohort. Now, I would like to explain how iPS platelets are produced. This slide shows the immortalized mesenchymal progenitor cell line, IMMK-CL system, the source cell of iPS platelets. IMMK-CL mesenchymocyte is established from iPS cells by the introduction of 3 transgene c-MYC, BMI1, and BCL-XL during mesenchymocyte differentiation and cryopreserved. In the presence of doxycycline, IMMK-CL expand. Upon depletion of doxycycline from the culture media, the cells mature and produce platelets. Next, we developed novel concept turbulent flow-mediated bioreactor system on IMMK-CL maturation and platelet generation by membrane shedding. In real manufacturing process, we prepared 4 8-liter tanks within GMP facility. This is the overview of the iPS platelet manufacturing process. From iPS cells, IMMK-CLs are established and cryopreserved as master cell bank. Upon demand, master cell bank stock is stored out and expanded. Then platelets are generated in the turbulent flow bioreactor as shown in the last slide. The produced platelets are separated, concentrated, washed, and resuspended. Finally, the package is irradiated. The whole process after master cell bank is performed within GMP facility. This is the actual package of iPS platelets. Similar to washed platelets, they are resuspended in bicarbonate ICD aluminum solution. The electron microscopy image shows iPS platelets are larger size, but have same ultrastructure as endogenous human donor platelets. The functionality of iPS platelets was confirmed in vitro and in vivo. In this rabbit model, rabbits were pretreated with busulfan and ethyl palmitate to induce thrombocytopenia. Experiment using research line iPS platelets shows the same hemostatic capacity as donor-derived platelets, depending on the storage time. In this model, hemostatic capacity of patient-derived platelets were confirmed as well. In most industrial countries, we have unbalanced situation between demand and supply caused by aging society and advanced medicine. In solution, we have done first in human study by autologous iPS platelets. Next, clinical studies by using allogenic iPS platelets will start soon, which is organized by startup company Mearcarium, collaborating with Sada Foundation and Kyoto University Hospital. Also, we plan to supply universal type iPS platelets by HA class 1 deficiency. Clinical evaluation of such product is planned in 2023. Mearcarium was established in 2011 by Professor Nakauchi and myself and other members. And now company is working for future commercialization. Our R&D is still ongoing with many collaborators and colleagues. Thank you very much for your attention.